viruses, fungi, and human DNA. This is what we're going to verify live in the next few minutes. You're going to see, this is the COVID-19 sequence. There's about, I think, 29,000 of these blocks of code. This is code. And that's what is published in WHO's website, and it's called the sequence of COVID-19. But in this sequence, we will demonstrate live here. You will see that this sequence has codes from known bacteria, viruses, fungi, and even DNA. You will see the consequences of this later on. So we have the microorganism that is being tested has little bits from the human body. That's what we're seeing here. Okay. According to WHO, COVID-19 has 4 specific gene sequences. So they've done all their studies around the COVID-19 virus, and what they came up with is there are 4 genes that are specific. So once you see those codes, you know, a code, remember, the sequence is a little bit of DNA. Nobody, no test can do the whole virus, you know, 29,000, that's impossible. You can only do little bits, 150 here, 500 here, you know, this is what you do. So they've said in the 29,000 sequence of the COVID-19 virus, there are 4 sequences, maybe about, 50 genes, not very much. But once you see them, you know you have COVID-19. And these sequences is what is now used in the kits. You know, they have all these test kits, all this testing. They don't test for the virus, the whole 29,000, that's impossible. You've got to test for a little bit. And this is what they're testing for. So they call these sequences, these codes, RNA polymerase gene, N gene, S gene, E gene. This is what they call them. And they are codes like the CT, GATT thing that we've looked about, this code here, right? So there are some of them in here that are specific. The minute you see them, you know you're dealing with COVID-19. And these are the sequences that are searched for with the PCR tests, right? So to detect COVID-19, you've got to add the sequences of these genes. This is the primer. So the primer has got to match these specific genes. You know, when you add the primer and the minute it changes color, you know, okay, there's N gene, this is COVID, or S gene, that's COVID, or E gene, that's COVID, or RNA polymerase gene, this is how it works, okay? So 4 of them, 4 specific gene sequences. But here is where it really starts to get incredibly troubling. I mean, this discovery deeply troubled me, still is. Like I said, you are going to do this test yourself. We're all going to become virologists here at the end of this. So what we find is like these 4 specific gene sequences that are used to identify COVID-19 are found in many bacteria and human cells. They are found in many bacteria and human cells. In fact, many of the genetic sequences of COVID-19, the 29,000 complete gene sequence of this microbe that they say is unique, that the sequences are found in dozens of other microbes and human cells. So basically, when they're doing the tests and they're testing for these genes, they are testing for human cells. They are testing for Staphylococcus bacteria, Campylobacter, all kinds of bacteria and viruses that share the same sequence. So there is absolutely nothing unique about COVID-19. When COVID-19 is being tested for, they're testing for when they put the swab down your throat, they're scraping some of your cells. Of course they will, right? They're collecting samples. Maybe you had a flu, or maybe you had a cough, that cough caused by a bacteria. They're also collecting those samples. And that's what they're going to test for in the PCR. And then they say people are COVID positive. We are going to demonstrate this here. That's actually the main thing. We're coming to the end of this webinar. I think I said, yes, I've got 3 minutes to go. So like I said, the primer, this is the the bits that you have to know to introduce into the PCR tool, because you've got to know what you want to look for before you use the PCR. So that bit, the primer, used to identify COVID-19 is found in many other microbes and human beings. And this is the code. I'll send you this. We're going to look at the COVID-19 code. Then we're going to look at the primers and probes approved by who. And now we're going to use the technology that virologists use to search for gene sequences. We're going to search for the probes and the primers that are approved. These are the things that are tested for. So we're going to search for these probes and see whether they match human cells or they match other microbes. We will do that with this website, and I've told you how to do that, and we're going to do it here as well. The COVID-19 PCR test is testing for DNA from human cells and other microbes. Do the search yourself. Do the search and see for yourself. Do you still believe in COVID-19? So that's what we're going to do next. I'm going to minimize this program, and then I will just open my browser, and then let's do the search. So this is a public, this is called Gene Bank. So this is where you go to see the genetic sequence of anything, any microbe, human beings, viruses, bacteria. You just type in the name, and then you're going to find a microbe. And this is the genetic sequence of COVID-19. It says so here, sorry about this, severe acute respiratory syndrome, coronavirus 2 isolate, Wuhan 1, complete genome. And that's the sequence. That's where it starts. And this is the sequence. This is how they list it, from the very first sequence, and then it keeps going on, 1, and then you have the ends here 60, it starts here 61, and carries on. And we can see here, we have 29,881 or something like that. So that's the sequence. So it's from this sequence that the World Health Organization, this is their website, this is World Health Organization, they have recommended probes and primers. They've got these from this sequence. They've got it from here, somewhere here, they got these. And they recommended, they say, this is what we have to test for. This is a primer. They have all different recommendations of what we need to do. These are the genes. This is for China, what they're testing for, the RNA polymerase and the N gene, these 2 targets in RNA. So they all have little bits of the specific gene. Each country is testing for something. So this is a primer. Just let's copy it. So what we're going to do now is to look at this, because this primer, like I said, is copied from here, somewhere here, right? And then what we'll do next is to go to the BLAST public domain. This is where you go to compare any sequence at all. You want to see if that sequence is found in anything else. This is where it's done. These are public domain sites. It's called BLAST, right? So I would, first of all, what I'm going to do is I'm going to see if that sequence is found in human. So I'll click on human, and then we can go to microbes later on. Okay, and now I would enter, just paste that sequence. And then what I'll do now is I'll make sure on that program selection, this program is going to show me the highly similar sequences, right? Make sure this is checked. Click on BLAST. And it's running the search. It takes maybe about a minute or so, depending on the length of the sequence. So this is the first one we're going to do. And we're going to see the result it's going to give us. So I encourage you, I will send the PDF with this link. I encourage you to actually, what I've been doing is just copying codes from here and running the search. This is very, very disturbing. So why does this have human genetic sequences from bacteria? The reason is those Chinese scientists, whether by, I don't believe it's carelessness. It's most likely criminal, right? They didn't filter the samples that they used to determine the code, the so-called code of COVID-19. So because they didn't filter the sample, they put it inside the PCR machine, and then the PCR, and then they started testing for sequences from all sorts, right? And of course, they were testing for sequences from human cells, from other kinds of bacteria, from all sorts in the samples. And because they also didn't have the original COVID-19, they didn't know what they were looking for. So they used a system called de novo assembly. So basically, they would now tell a software, this is the realm of artificial, just nonsense, not real. They would put this something, a code inside this, one of the codes that they've got from the PCR, they put it inside this software, they run the software, and then the software would now kind of like put the little short links together end to end to build this thing, right? And then of course it's going to build with all the codes in there, all the bacteria, viruses, human cells, and then that's why you have 29,000 base pairs, and it's all full of sequences from all kinds of other organisms. So let's look at our search and see what we've found. Now we've done our search, it's completed, and this is what we are finding, that the primer, what it's been tested for. So when they take a sample for you and from you, and maybe that lab is using this primer, it could be any of them. We're going to do a few tests here. It's using this. So this is what they would use. They would tell the PCR machine to look for this inside your sample. But this is what we're finding here, that the primer, I don't know how to get rid of this now. Right. It contains human cells. Human cells. This is it. Human. 100% identity match. All kinds of chromosomes in humans. This is what we're looking for. This is it. So when they do the test, they're actually testing for chromosomes from your body. This is, and then when they find something from your body, they tell you you are positive. That's what they're doing, right? Let us copy another one and now check for microbes. Just like basically we'll copy them at random. I haven't done these. I've done quite a few searches here. I haven't done a lot. Now let's look at microbes. So you have your cough, normal cough, you have fever, which lots of bacteria, fungi would cause. You go to the hospital, they suspect you of COVID, they put inside the PCR this primer, and then let's see what we're going to get. So let's see if there are viruses or bacteria or microbes or other things that would cause fever, would cause cough, would cause sickness, that share the same sequence as what is being used to test your body for COVID-19. We're running the search here. I encourage everyone to do this search. I have, I kind of discovered this a few days ago. I've known about the PCR, but actually taking the time to do the search myself. I think many weeks ago, there was this woman that first alerted the world that some of the sequences that were in the coronavirus were also found in human beings, so it's kind of taken us quite a while to really start to look at things properly, to look at the sequence, to now do the search ourselves to find out what's going on. So we've completed that search, and this is what we are finding. We are finding that sequence, right, that they're using to test for COVID from the swab sample, these are all the microbes that share the same. Like this is probiotic, lactobacillus, right? These are bacillus, these are microbes. A lot of them I don't know of, but they're there. Most of them probably wouldn't be able to cause diseases. Mycoplasma, some of them you recognize, E. coli, right? E. coli. So you have dysentery, right? They do the test. It's going to come up positive. Look at it. probiotic. Maybe you take a little bit of probiotic that morning and have a little bit of fever because your body's trying to heal itself. You go for the test, that's it. You're positive. These are all the bacteria that share, and these ones are incredibly high query rates, 89% match, absolutely high matches, right? Very, very similar. This is what they're picking up. So, well, that's actually what I really wanted to introduce everyone to the science, because the media is totally blacked out any kind of discussion around COVID-19, any kind of intelligent discussion. Nothing is encouraged anymore. And it's got to a point where we all have to become scientists, because you see, what is going to happen is they are basing their vaccine on this code, right? So they are building something that is anti this code, anti-codes inside your body. They're going to inject, they're producing vaccines that contain the opposite. That's what a vaccine is. A vaccine would want to destroy the virus, right? So it's got to destroy these codes. And a lot of these codes are your human genetic code. So the vaccine, it's impossible that the vaccine will not harm the human body. But I think it's even going to do a lot worse than harm the human body. The vaccine is going to transform. This is a time that I never suspected, that such a thing could happen. I knew that things were not right. Medicine. That's why I practice natural medicine. There's all sorts of awful stuff that goes on in mainstream medicine. And I realized this very early on and decided I was only going to do the right thing. But to imagine that the corruption will be taken to this level, it's unbelievable, right? Where they are banking on everyone being too stupid to think logically, right? They have introduced a disease that doesn't exist, a test that isn't even used for what it's not even meant for that purpose, the PCR. And even if you want to imagine it's meant for that purpose, it's actually testing for human cells and other kinds of microbes, many of them harmless in the human body. And they've declared a pandemic and they've instituted this change. I mean, look at what has happened to humanity since the pandemic was declared. We've gone into global lockdown. They have banned people from socializing, right? Their so-called guidelines, what they're advising us to do are steps that would further isolate humanity. They're pushing us away from each other. And we know that isolation, there's actually a study, I'm going to find that and attach it, that was done many years ago. Very, very extensive study. And they were looking at the factors that put the most pressure on elderly people, factors that would literally, have the capacity to cause the most disease in a person. And they found it was isolation. When you isolate human beings, we die. We are social animals. And then you find these measures are measures that are telling us that we are bags of diseases. We should cover our face